when Xiaodong Wang at Emory University showed that a protein that I spent almost working every day with is cytochrome C. And it's the substrate for cytochrome oxidase. I got my PhD in cytochrome C oxidase kinetics, looking how it changes with oxygen. And cytochrome C is a substrate for it. And all of a sudden, he had this a landmark paper which showed that cytochrome C, if it's released into the cytoplasm, it activates the caspase 9. And you have to just sort of go back a little in time to think about that, what does that really mean? Because if you're brought up in the previous century, mitochondria make ATP, maybe they also sustain growth by making metabolites. That's a simple way to think about it. And really today, what's happened in the last 25 years is that mitochondria release molecules that could interact with the rest of the cell, right? So cytochrome C with APAF and caspase 9 for cell death, peroxide release to control gene expression, things like NF-kappa B. You have, oh, there you go. Okay. Now, yes. All right, now I can go back and present this. All right, we'll make up in time real quick, I promise. And so, you know, okay, so really the way to think, this cursor's not working. Okay, so you know, the fact that cytochrome C, which is part of the electron transport chain and is used by cytochrome oxidase, is released, is to cause cell death. I think that was, at least for me, was sort of a aha moment in the sense that a protein that actually, to be honest with you, this is a true story. I told Craig Thompson, who told me about it, I told him it was an artifact. I told Xiaodong Wang years later that I thought it was an artifact because it was so hard to imagine that, you know, you're having this protein that gives you life in ATP, but if it just gets released in the cytoplasm, executes a cell death pathway. And I'm not going to go through all these. Of course, you know, Jerry Shadle here was pioneered mitochondrial DNA release, and Michael's done some really fantastic work on linking it to immune receptors like NRLPR3. These are just the signals that I care about. That's the ones I put in. There's a whole slew of other signals from calcium. And then beyond the signals released, you know, the mitochondrial outer membrane, as Susan has shown, can serve as a platform for a lot of signaling cascades. So I think this has now come of age. And this is where you can fall asleep after this. If you ask me why mammalian cell respires, I would have told you bioenergetics, they need it for survival, although in many systems, glycolysis can sustain ATP for survival. The other one is really through our cancer work, we've shown over and over that it supports growth by generating the macromolecules. And the last one is signaling, and all these signaling cascades, which can then dictate cellular fate and function. And this is really what I want to today highlight with two different examples. And so the first example is this unpublished story on lung development. I think we can all agree the lung has become the center of attention the last 2+ years during the pandemic. And really in the last 10 years, the lung field has been sort of revived with molecular tools and showing how you know, the lung epithelial cells generate a healthy alveoli. And just to remind you, there's type 1 cells. These are the gas exchanging cells. Type 2 are the ones that make surfactant. And the two together sort of make an adult healthy alveoli. Of course, there's many other cell types in there, including endothelium to bring in the blood vasculature. And in steady state, during a lifetime, the type 2 cells can serve as a reservoir, they self-renew, and they can make type 1 cells sort of like a stem or a progenitor pool. And what ends up happening is that if you have things like SARS-CoV-2, influenza, asbestos, it'll damage the type 1 cells. And once you have a damaged alveoli, what can next happen is you have two choices. The damaged alveoli can repair itself and go back to homeostasis, or you can get fibrotic lesions. That's what has been asbestos, or you get really bad respiratory failure and you never get off a ventilator. And unfortunately, the patient dies, right? And so what we wanted to do was ask what's happening to mitochondrial function during development largely. We've started to do post-development as well. And one of the first things we did is we just mined single cell data that was published. This sort of paradigm happens across many organs. So hearts are probably one of the better examples, where what you have is over time from E12 in mice is till P14, there's sort of a slow down regulation of glycolytic genes and an up regulation of oxidative phosphorylation genes. And this makes sense that, you start to see a transition as the embryo becomes less and less hypoxic and more differentiated. It needs to sustain, most people think, just the energetic demands of differentiated cells. And that's why it's happening. And of course, that's the simplistic canonical way to think about it, especially as you develop, you switch to a fatty acid oxidation metabolism that sustains the health of a heart or a lung or a brain, right? And again, this is all about sustaining bioenergetics, a way of thinking about it. Now, what we did is some genetic experiments to see what would happen. And then one of the things we did is we went in the electron transport chain and we wanted to knock out mitochondrial complex 1, specifically in lung epithelial cells early in development. Now, what are the predictions? Based on what I just showed you, the predictions would be maybe you get an alveoli made, but it dies very quickly right after birth because that's when OXPHOS needs to sustain the health of the alveoli. Right, that's one. The second thing is during the process of development, you have proliferation going on. And as I said, mitochondria can sustain proliferation by preventing metabolites for growth. Maybe one thing would be that, you just don't make it to development, make it to right postnatally, right? So either you don't make it to postnatally or you die after postnatally, or you're okay for a while, but something else happens where the signaling cascades sort of determine cell fate or cell identity of these and so that's how going in we thought about it. And the way we did this is we took NDUFS2 flox alleles. So this is a core catalytic subunit of mitochondrial complex 1. Mitochondrial complex 1 has 45 subunits, and this is one of the core subunits. You knock it out, you can remarkably decrease the activity of the complex. So, and this is the recombination happens at 10.5. In other words, you knock out mitochondrial complex 1 in alveolar epithelial cells largely at day 10.5, and this is complex 1, and you knock it out, so the electron transport chain falls apart. Okay, so what happens? Well, the mice are born, and that was a little bit of a surprise. The mice actually live till a couple of weeks, up to day 50, 6 to 8 weeks is before they die. And really the first 2-3 weeks, by histology, they look remarkably similar to the wild type, and then they progressively get worse, and their compliance gets worse, and they look like, they're dying probably of respiratory failure. Okay, so that was, good. Mice die, means you have a phenotype. The other thing is, they do largely develop, right? Which is also kind of interesting because now you can get cells out, you could do some mechanistic studies, because if they would have died in utero, that becomes difficult, or right after birth, but they actually sort of develop for a while and then they progressively have this pathology. So you can do some mechanistic studies. So one of the ones we do, which I think is quite interesting, this is mitochondrial complex 1. There's 45 subunits from 2 different genomes, mitochondrial DNA and nuclear DNA, and they have to come together to make this large complex. The large complex proton pumps, and this proton pumping is important for ATP synthesis. It also does NADH to NAD regeneration, and that keeps the TCA cycle moving on. And these 2 electrons are donated to the downstream complexes, right? So there's a protein called NDI1, which was pioneered by Professor Yagi at Scripps here in San Diego. This was one of these papers published in 1998 during my postdoc years. Always stuck with me back there because I thought it was a super cool paper. What he found was these proteins, NDI1, and there's a variety of them, they could just do NADH to NAD and pass the 2 electrons on to ubiquinone and down the electron transport. So in many ways, it was a bypass system. It just didn't do proton pumping. but it was one subunit. Right? And it would bypass inhibition of complex 1 in cell culture. So we generated a conditional overexpressing version of NDI1. So this is where and by the way, the NDI1 is from yeast, right? So this is genetically optimized, the codons for human. So you have this lock-stop blocks in the ROSA locus, NDI1, and you have your flox alleles for complex 1. You take the surfactant protein C at embryonic date 10.5. Whenever you lose the NDUFS2 flox, the complex 1 activity, you have NDI1 come on, right? And then your chain looks like here at the bottom. Complex 1 is gone, but NDI1 is there to take NADH to NAD, keep the TCA cycle going, and you pass electrons to Q and down to the rest of electron transport. It's like a bypass system. Now, what we're asking in vivo, think about this for a second, 45 subunit complex that falls apart due to NDUFS2 loss. And we're asking a single protein in yeast, an ancient enzyme, to come in and rescue that function and in the cells in vivo and hopefully the mice. And so the first thing we did is before the conditional NDUFS2 knockouts died at day 35, they have high amounts of lactate. And that makes sense. You lose complex 1, you should make more lactate because you're glycolytic. The NDI1 completely rescues this. I think we do. At the same time, the way we've done this is, we can put a reporter also to look at efficiency of the Cre. But we can take cells out, the epithelial cells. In the knockout, you see all these changes, right? Here's the NDI1 rescue, though largely many of those changes go away. It just tells you that the NDI1 is having and restoring metabolism of the knockouts back to the wild type state. Complete rescue, right? So on one hand, you can say a little, maybe expected, right? Because you're fixing the metabolic defects of mitochondrial complex 1. And on the other hand, I think it's pretty cool. You can take 45 subunits and you can rescue it with 1. It makes you wonder why we need the 45 subunits. And I'll get to that later, right? Why don't we just have this yeast version sitting around in us? Why this complicated 2 genome system of, but at least in this particular cell type in utero, putting the yeast complements this. So one interesting thing that many of us do is obviously single cell. And I think the developmental biologist, I think the single cell is great for anybody doing developmental biology because you can really sort of study what's going on. And what we know is here are the type 2 cells, and they eventually go through a transitional cell state to make type 1. And what we can see is the knockouts really have a lot of transitional cells compared to the control cells. So like the control cells have some type 2 cells, and then they have some type 1, but the knockouts really is where we saw a lot of this transitional cells. And in fact, because we have a reporter, or with wherever the SPC is positive, the reporter lights up. And as you can see, the transitional cells also are lit up for the reporter, okay, the TD tomato reporter. That just tells us we think our transitional cells are coming from an SPC positive cells. So those lung epithelial cells was where we had the knockout. And this is a quantification, and where you can see there is a sort of an increase in a transitional cell population in the knockout. And that was exciting to us because it wasn't like, it's really suggested there was a cell fate or a cell identity issue now, or that mitochondrial complex 1 was controlling, right? You go from type 2 to a transitional to a type 1, and all of a sudden you're stuck and you have this accumulation of these transitional cells. And the other thing you could start to do is mine to see what kinds of gene expression you really see that's elevated. And one of the top signatures we saw was the integrated stress response. And in fact, in many of our studies in vivo, when we look, you could do it in Tregs and T cells and many, many systems. This is one of the top signatures that comes up over and over and over. And just to remind you, the integrated stress response ultimately downregulates protein translation, with selectively allowing certain transcription factors, particularly ATF4 and ATF5 to be activated. And there's been quite a bit of work now done linking mitochondrial dysfunction, and that in other words, if you give mitochondrial stressors, mitochondrial inhibitors, there's either through GCN2 or HRI. And the GCN2 seems when aspartate and asparagine levels go down. The HRI is a little mysterious how that's triggered. And I can tell you in our system, those metabolomics data that I showed you, aspartate and asparagine are not down. And in our knockouts. So we think it might be more HRI, although we have to genetically confirm that's what's driving it. But before we do any of that, is the ISR necessary for this accumulation of transitional cell and this developmental blockade? And so there, Peter Walters, many of you know, has this wonderful compound called ISRIB, the integrated stress response inhibitor. Remarkably, they've tried this from Alzheimer's to ALS to a variety of diseases, including cancer. David Rougier had a nice paper using ISRIB to prevent prostate cancer metastasis, if I recall. It works quite well. It's they figured out structurally how it works, et cetera. You can read up. There's lots of papers on it. So we dosed our mice. And this work, by the way, was done in collaboration with Sung-Hae Han. And Sung-Hae basically give ISRIB starting at P11 and until P100. And you can see remarkably, many of these mice are rescued, right? So think about it. ISRIB doesn't fix that mitochondrial complex 1 activity, right? The NDI1 definitely did. So you could say, well, that makes sense. But this is coming downstream of the complex 1 deficiency. And remarkably, we're seeing, based on unbiasedly the gene signature, right? And obviously, this is now correcting in the lung pathology and allowing proper differentiation to occur. And these mice look quite healthy. So one thing that came out of this is, mitochondrial complex 1 and the NDI1 rescue really pinpoints to the NADH, NAD as a pivotal point in driving the disease. So complex 3, 4, 5, all of these downstream, if you were to inhibit it, you'd get a similar phenotype as complex 1. And in fact, when we knock out complex 3, you get quite a dramatic phenotype. Complex 2 is interesting because it's on a parallel pathway, right? In the sense that both complex 1 and complex 2 donate 2 electrons to the Q pool. And complex 2 doesn't affect the NAD. So if you were to knock it out, it will not affect the NADH, NAD regeneration. It'll make these cells become glycolytic, et cetera, and make the TCA cycle fall apart. But what would happen, right? And I thought this was an interesting experiment that Sung-Hae did. And again, apples to apples, right? The same surfactant protein C, Cre. We're using SDHD floxed alleles. Here's you knock out complex 3. Complex 1 is still able to regenerate NAD, but because complex 2 doesn't work, the TCA cycle falls apart. Right? And you can see that here. Here's complex 2. If you lose complex 2, succinate goes up, the TCA cycle doesn't work, oxygen consumption goes down. You can measure succinate levels get very high here in the conditional knockouts of SDHD. And then, of course, what would happen in vivo? And remarkably, not much, right? So these are also glycolytic. Their oxygen consumption is down, and yet they have a completely distinct phenotype in the complex 1. Now, the compliance and the survival argues that they're remarkably healthy, and they are. The pathology indicates they're not totally normal, right? They're just not, their pathology is a little messed up, not as severe as the complex 1. And so it's on a spectrum, right? They're not quite like wild type, but they're not as pathogenic as the complex 1 knockouts. They're somewhere in between where their histology doesn't look proper, but it's not so bad that it compromises the health of the mice or the lung compliance, the functional outputs. And this you can see here because this is the integrated stress response now. And you can see the NDUFS2 really have a marked increase in ATF 4 and 5 compared to the SDHD. So the SDHD null, and this is an integrated stress response score right here of all the genes, that there is, compared to their wild type, you can see some increase clearly of the ISR signature, much more robust between the NDUFS2 knockout. This is the NDUFS2 knockout compared to its control. Much more robust in the NDUFS2 knockout compared to the SDH. So, again, I don't want to say that the SDH knockouts look like the wild types. It's on a spectrum. The wild types are the healthy controls. The SDH do have an ISR, but it's not to a level that is pathogenic. And the NDUFS2 have a very robust ISR signature. And as you know, if you give ISRUB to the NDUFS2, we could start to rescue these mice. And what this sort of argues is the following, and I think, Michael, you would agree with this based on the NF-kappa B literature. I learned this in the old days working on HIF. NF-kappa B, HIF, the ATF4s, the ISRs, they're all designed to come on and then come off, right? They're designed when you have a stress, and they're there to help you adapt to that stress, but they are supposed to come off. And many of them are tightly regulated by ubiquitin ligases and half-lives of quick protein turnovers. I think we can agree on that. But just like in the HIFs, if you lose VHL, it's negative regulator, and you have constant HIF activation, then it's bad for you, right? You get pulmonary hypertension, you get renal carcinoma, all right? So chronically high will give you pathogenic. And we're proposing the same thing here, where the chronic elevation of the integrated stress response is pathogenic. And that's what we're seeing in the NDUFS2. The SDH is chronically low, not at a level that it gives you pathogenicity, but you can live with it, right? And really the model is that, again, mining the developmental single cell data, you go from a type 2, a transitional cell, to a type 1. There is an adaptive ISR signature during normal development. Again, HIFs, NF-kappa B, the ISRs, all these pathways are supposed to come on and off during development. And of course, when they reemerge in the adult as chronically activated, they cause pathology, right? And so what's happened here is that the mitochondrial complex 1 increases NADH, NAD, and we fix this by NDI1. And this triggers a pathologic ISR, because if we rescue this pathologic ISR by giving the ISRUB compound, we can bring that down to a normal transitional cell state in type 1 cell. But if this chronic high NADH, NAD ratio gives you this ISR, you accumulate this transitional cell and you don't get differentiation to type 1. I'm going to stop there and take a question. Yes. So the question was, so these cells are relying largely on glycolysis and glucose? Yes. Yeah. What's interesting is both the SDHD knockouts and the complex 1 knockouts. So both the complex 1 and complex 2 knockouts, so when we look at their oxygen consumption rates, they're markedly similar. They're down. In other words, they have impaired respiration equally, but they have a very different. One has a very chronically high, the complex 1 knockouts, and those mice die. The complex 2 have a chronically low ISR signature and they live. So that's sort of the model, working model that we have. And I think what it shows is beyond cells dying, cells not able to grow, because these transitional cells that are accumulating, they are proliferating, right? And that really it's an issue of cell fate and cell identity that's being controlled specifically by mitochondrial complex 1, NADH, NAD, funneling into the ISR and a developmental program. And I think this is it's a nice way to highlight that mitochondria beyond bioenergetic and biosynthetic is controlling cell fate or cell function. So let me, I'm actually doing reasonably well on time. Let me just switch to, you have to pan to your host here. So I have to talk a little bit about inflammation, which I know very little about, but I read Michael's papers and others in the field. And mitochondria have been connected to many aspects of both the innate and adaptive immunity. And a couple of things, there's TLR4 signaling and ROS as to interferons and NF-kappa B, of course, mitochondrial DNA for TLR9, CGAS, NRLPR3 as well. And both ROS and mitochondrial DNA are inputs into the NLRP3 inflammasome to activate caspase-1 and IL-1 beta, right? And really what I want to talk more today about is NRLP3, which has really come of age, right? One of the reasons people are very interested in it is activated by what's called sterile inflammation, right? And it's not only driven by pathogens, but whether it's beta amyloids to gout for crystals, and so it's obviously people are thinking of targeting it for chronic diseases. And so let's just focus on the inflammasome. As many of you know, I don't need to introduce this. Typically what you do is you prime with LPS and you get all the inflammasome components upregulated, and then you can give things like ATP or nigericin. Potassium efflux tends to be one common element to it. But there's clearly lots of work nicely showing that both mitochondrial ROS and mitochondrial DNA are also inputs into the system. And eventually you get caspase-1, you get IL-18, IL-1 beta from as well as gasdermin, which then causes pyroptosis to release these active forms of these cytokines, right? So we had, Michael had sort of seen this nice link of mitochondrial DNA and oxidized form of mitochondrial DNA. And we gave him our TFAM mice, which he was able to really nicely show that they wouldn't have robust inflammasome activation, and you could reconstitute by giving back the oxidized mitochondrial DNA in the absence of TFAM, and TFAM generates mitochondrial DNA. I got into it for a different reason, which is mitochondrial ROS, because we've done a lot of work on it. And the problem with the mitochondrial ROS theory was, and Michael, you can correct me on this, sometimes antioxidants worked, sometimes they didn't, sometimes ROS would activate it, it wouldn't activate it. And then just stepping back from it, the other question was, well, what about electron transport chain, right? If you inhibit it, some people, the late Jörg Schaub reported that rotenone would activate it, which is a complex 1 inhibitor. Others showed complex 1 inhibitors maybe turn it off. Others show, like Gabriel Nunez, mitochondria have nothing to do with any of this. And these are all very prominent people. And I think in retrospect, I mean, the TFAM is nice in one case because the TFAM, there's a genetic strategy, right? There are flox alleles you can make. And I think if you were testing directly electron transport chain, and you should do some of the models that we've used in the previous study, right? The complex 1 loss, et cetera. And so we just step back and literally just ask one simple question, and this will probably be my one and only study on the inflammasome, because I'm somebody who's fundamentally interested in electron transport chain function. Everybody's got a favorite kinase they work on, PKA, mTOR, et cetera. ETC is like my kinase, right? This is what I perturb. Downstream of it, I'm trying to find functions for it, right? Is it all about generating ATP or ROS, et cetera? And so we basically didn't reinvent the wheel. We did exactly what other people in the field do. There's a priming phase where you give LPS, and then you give ATP 10 minutes later, or you can measure caspase-1 activation. 30 minutes later, you can measure IL-1 beta in the supernatant. These are bone marrow-derived macrophages. One of the first things we noticed remarkably was when we gave any metabolic ETC inhibitor at this time point, we didn't see much differences. It wasn't a good activator, and it didn't inhibit it very robustly. So then we gave it prior to LPS, and then we started to see, and I'll show you the effects. So all the inhibitors are given here. One of the first things we noticed, and I'm not going to show you, it didn't really affect, again, these are short time points, right? Sometimes people use these long 24, 48 time points with these metabolic inhibitors. These are relatively short couple of hours. We didn't see any changes in the inflammasome components. So all the priming effects were not inhibited by any electron transport chain inhibitors. As the effects were all seen when we looked at caspase-1 activation and IL-1 beta. So that's important to note. So the other thing we did is most of the data I'm going to show you is caspase-1 and IL-1 beta, which are the downstream effect. So one of the things we thought about doing was, instead of specifically using the knockouts, which would then maybe rewire the cells forever, right, from the beginning if you lose complex 1, maybe we thought about using sort of like a pharmacologic / genetic combination. What do I mean by that? And so here's a wild type mice, and it's known as, when you give LPS, succinate increases. And one model that Luke O'Neill and others have suggested is, when you have this high level of succinate, that you can get a lot of ubiquinol generated, and that can go back to complex 1 and generate superoxide. So one of the things you can do, and if you give complex 1 inhibitors, it can prevent inflammasome activation. And the one that we like to give, and is not as around, known as pyrsidin, is much more. But we had a good genetic technique to show pyrsidins on target effects, because we can overexpress NDI1 in our bone marrow macrophages conditionally. So the idea is, if you give pyrsidin in the presence of NDI1, the endogenous complex 1 cannot generate any ROS. It's only because of the NDI1, it's going to go in the forward direction, never in the back reaction. And then you can see what happens. All right. So you can see pyrsidin, 100 nanomolar, 500 nanomolar decreases oxygen consumption rate. With NDI1, you completely rescue it, right? But if you look at caspase-1 cleavage, it's down, but the NDI1 completely rescues it. Right? And you can do this with IL-1 beta. IL-1 beta goes down, NDI1 completely rescues it. Right? And so, because the NDI1 looks like this with pyrsidin, so when you give pyrsidin, you block a complex 1 from doing red and making superoxide production. I mean, the biochemists like myself have shown this in many in vitro assays, but the presence of NDI1 just moves everything in the forward direction, and you rescue everything. So it doesn't seem like complex 1 generated superoxide is an input. Okay. So then one other possibility is, maybe downstream of it, complex 3 generates superoxide. In fact, a complex 3 can generate superoxide in the intramembrane space, and that can become H2O2 and leak out quickly to the rest of the cytoplasm. So we could start to perturb that as well. So here's the wild type state. There's an inhibitor called mixothiazole, which is known to inhibit mitochondrial complex 3. So like pyrsidin, it's quite specific. But now we can do some fancy little tricks. Like, so we've generated conditional knockouts of mitochondrial complex 3. These are QPC allele. It's just another subunit of complex 3. That'll get rid of the endogenous mitochondrial complex 3. We use SFTPC-Cre. But like NDI1, there is a rescue, and the rescue is something called AOX. It'll take electrons. So normally electrons from ubiquinol go to 3 all the way to oxygen as the final electron acceptor. Ubiquinol donates electrons to AOX, and AOX will then donate those to oxygen. It's a bypass system for complex 3 and 4, right? The one thing is, again, when you do this system, you really decrease H2O2 production, not completely, but markedly. Right? And again, OCAR goes down in the complex 3 knockouts. With AOX, it's rescued. Right? Exactly what how you would design the system. You can look at rate of H2O2 production. It goes down with the QPC knockout. With QPC AOX, it's not markedly recovered, because again, in this system, you're not generating as much H2O2 or superoxide and then H2O2. So it's a good way, again, and many other people have shown that AOX is a good way of reducing ETC generated superoxide. So what happens? So again, if you look at caspase-1, it's down. And this is with mixothiazole, which is the complex 3 inhibitor. Again, with mixothiazole in the complex 3 knockouts, with AOX, it's all rescued. really arguing again, it's not probably about H2O2. Now, we used a lot of mitochondrial targeted antioxidants. We gave mito ROS agents like mitoparaquat. Nothing made a difference. And I think one sort of soft conclusion for us is that we don't think mitochondrial ROS is a heavy input into this, at least the ROS that has to be released. Again, we can look at IL-1 beta, it goes down, it's markedly rescued. In vivo, the complex 3 knockout with the AOX, in vivo, these are the low ROS state, right? They still generate IL-1 beta in response to LPS. Yes, and so again, we think that mitochondrial ROS is not required for inflammasome activation. Here's complex 3 knockout, AOX again. In the wild type state, you have all these places you can generate at ROS, and really markedly a lot of this is reduced. It's not completely down to 0, so you can imagine you can still maybe get a little bit of ROS to oxidize mitochondrial DNA for its release, but more that H2O2 is leaving and maybe activating a kinase or something that, or there's this idea that the cysteines and NRLPR3 are being oxidized. I'm not completely convinced that's going to hold up, at least not the ROS coming from mitochondria, maybe from other sources. So we gave all these inhibitors, they all do it. They all inhibit the inflammasome. And they have all sorts of changes in metabolomics. The other one we did is, sorry, I think this is my computer change, but if you give oligomycin or FCCP, which make the potential go, mitochondrial membrane potential go up or down, inflammasome goes down. It didn't matter what sort of mitostressor we gave, and all of these are known to the biochemist, some increased ROS, some decreased ROS, some increased membrane potential, some decreased. They all did it effectively, right? So what's going on? So this is where metabolomics helped us. In all these conditions, so this is dimethyl malonate, widely used, that has in vitro and in vivo, it inhibits inflammasome. Luke O'Neill's nicely shown that. And we can look at metabolomics here. This is mixothiazole. Sorry, dimethyl malonate is complex 2, mixothiazole is complex 3, oligomycin is complex 5, pyricidin is complex 1, FCCP is a protonophore. A common metabolite was phosphocreatine. This was one common metabolite which was diminished with all these inhibitors, right? Each inhibitor had its own signature. This was the common one. Right? It was depleted. So just to remind you, when mitochondria are generating ATP, some of that excess ATP is put on, the phosphate is put on creatinine, and you just bank the phosphocreatinine, right? If ATP is like a currency, you're using ATP like the money all the time, and the phosphocreatine is like putting it in the bank. And of course, the muscle does this all the time. So when you run, there's a buffer there to help sustain and metabolism. Long time ago, it was shown that cyclocreatine, which kind of looks like phosphocreatine, is used in people, can deplete phosphocreatine levels. So here's cyclocreatine. It'll deplete phosphocreatine. You can look at the phosphocreatine to creatine ratio is markedly down when you give cyclocreatine. So cyclocreatine is a way to deplete phosphocreatine levels, right? And when we did this in vitro, you can markedly reduce the caspase-1 activation and the IL-1 beta. We didn't have floxytyleals for this for the enzyme and that passes on phosphocreatine to ATP. So we used that, but that again, the IL-1 beta levels were down, right? Mimicking the cyclocreatine effects. In vivo, when we gave this in the drinking water and gave it IP, the cyclocreatine decreased IL-1 and beta levels, right, in the serum. Right, suggesting there's a necessity of phosphocreatine to get this to sustain inflammasome activation. And in the way we're thinking about it, it is that, when during the LPS, we clearly notice that phosphocreatine levels go up because there's quite a bit of ATP that's being generated and is being stored there. And when you give an inflammasome activator like nigericin and I'll show you in the next slide, that ATP from phosphocreatine, or that phosphate from phosphocreatine, helps sustain the ATP levels to keep the inflammasome activation. one of the things that, again, is sort of these dogmas that, you know, when you give LPS through TLR4 signaling, you have a lot of HIF and glycolysis. But one of the things we notice is as soon as you give nigericin, you get this steep decrease in oxygen consumption rate, and you have this elevated in ECAR as well. When we give pyrsidin, that complex 1 inhibitor, OCAR is always going to be down, but those cells have high levels of glycolysis, right? So if you give pyrsidin, you don't make ATP, phosphocreatine goes down, you're purely glycolytic, but yet that glycolytic ATP is not sufficient to sustain the ATP demand of the NRLP3. And you know, there's these nice papers, and here's just two, where you know it's known that the Walker domain of NRLP3 requires quite a bit of ATPase activity, right? And so ATP hydrolysis is a feature, and I think that you must need some localized amount of ATP, which glycolysis can't support by itself. And when you give pyrsidin, and you're having a double whammy. You're not making the ATP from mitochondria, and yet you don't make enough phosphocreatine either, because you for that small amount of time, we think you need enough ATP to sustain an NRLP3 inflammasome. And so we think one input, to our surprise, is just, you know, we don't think it's ROS. Mitochondrial DNA could be clearly one input, but the other input is just ATP. And I think Michael had a nice paper in Immunity where they alluded to this point, the necessity of mitochondrial ATP for inflammasome activation. And we're linking this to also to phosphocreatine levels. And it's known that cyclocreatine can have immunomodulatory effects. I think people have now generated a variety of knockouts, floxed mice, and they're looking in T cells and macrophages. And I think you'll see more and more biology of creatinine and phosphocreatine in controlling innate and adaptive immune responses. And this paper should be out soon. Anyways, I'm on time. It's 12:51. I know we started just a little bit late, enough time for questions. So Leah Billingham did that nice study that on inflammasome with help of Josh Stuhlman, Jason Miska, and Taylor. Sung-Hae Han, who's a ND, who took care of a lot of COVID patients and managed to find some time to do that beautiful work on cell fate and cell identity. She's a real hero of mine, and she's going to really take this work forward. And Scott Buttinger, who's the chief of pulmonary critical care, also doing a lot of COVID research and knows a lot about the lung. Jose Lopez Bernal has been very kind in giving us NDUFS2 and SDH flux. We have a nice collaboration. Dr. Lee's lab and Young Jin, especially, who's done some of our single cell analysis. And I'm very grateful to them for all of this. Michael, I'll thank you for the invite, and others that I'll meet later, and people I've already met today. Thank you for your time. I'm happy to take questions. I think we have some time. Yes.
So the ATP is required for inflammasome activation, but how do we know if it's active in the mitochondria,
right,
where we think it's required to provide energy for gamma,
right,
or X?
Yeah.
Is there a way to separate?
I think the best way is the cyclocreatine, right? Because that still would allow the mitochondrial, the mechanism you proposed, because the ATP is being generated, but the transfer to phosphocreatine has already happened, and that can't go down to the ATPases. So I think that's one way to do it. And to be honest with you, just like you, whether it's the enzyme, what is it, CP, the enzyme that you knocked out, along with TFM, the enzyme that no, the I'm having a senior moment. The CMPK2. Right. So all that ATP requirement is still, if you really inhibit mitochondria from making ATP, you'll inhibit that reaction and the phosphocreatine. So I think the cyclocreatine gets to that. I'll be honest with you, we're very confident about our ETC data, right? Because we've rescued it beautifully with all this stuff. The best data we have is cyclocreatine, and take it for whatever, some siRNA. So, take it for what it's worth. I wish I had floxed alleles. As you can see, I'm really wedded to match genetics or using pharmacological tools that are rescued. But cyclocreatine, has been widely used, and pharmacologically it's been well studied in vitro and in vivo for a long time. So, Susan.
I'm trying to remember my biochemistry. How is creatine kinase regulated, and there are different isoforms?
Yeah, there's tissues. Yes. So how is it regulated in macrophages compared to the other ones? We haven't done a deep dive in that space. What they do have is a way to transport creatine. So many of these macrophages, the creatine transporter is really highly upregulated, especially like in tumors and in other places as well. Also, I think there's more to come on that. And there was a nice paper in Cell Metabolism where they knocked out that transport in macrophages, and that could have immunomodulatory effects. So I think that's the way to start studying it, not only at the enzyme level, but look at those transporters. No, sorry. This is the plasma membrane. So you get the creatine. Yeah. Great. Yes. Yes. Yeah. Right. Yes. Excellent question. Very, very astute question. So as you know, complex 4 pumps 4. The other ones make the other 2 and 4. So you get about 10 protons that are being pumped by the ETC. Now you have 4 gone. So 40% decrease. That should be a huge bioenergetic crisis. It's when we've looked at coupled respiration, no difference with the NDI1, because the other complexes clearly are able to sustain it. And that's what we've noticed. What I find more remarkable is the AOX, because AOX gets rid of complex 3 and 4 proton pumping. So 60% is gone. And yet complex 1 can sustain in coupled respiration. That's the respiration coupled to ATP generation. And so there is, it's remarkable they're able to compensate. I will say, I didn't answer my own question as I sort of rhetorically posed it. Why don't we just have a yeast version? Right? Why do we need this messy 45 subunits? So it turns out in macrophages, doesn't matter. In alveolar epithelial cells, it doesn't matter. But in some brain cells, it matters. Right? So there are some brain cells that we found out, you knock out complex 1, you become ataxic, you don't rescue it. You can rescue their lifespan, but you don't rescue the ataxia. So I think, I think it'll be in the brain. And there may be some other cell types as well. So clearly, I want to just pause real quick. There are 2 questions for the people in the chat. Jane Smitham is asking, did you measure H2O2 ETC to confirm? Yeah, we did. We showed you some data where in the AOX, the H2O2 secretion from the mitochondria is markedly down. And that's consistent with what Mike Murphy showed in a very nice JVC paper. Kunling Guan. Great. Do type 2 cells develop normally in the lung in the complex 1 knockout young mice? Yeah, so that's a good question. What we do see is some type 2 cells, those that are there, but we see a huge increase in type on these transitional cells. Now the question is, are those type 2 cells completely normal? Well, but they just go to the next one, transitional cells, and it's the transitional cells that are really now stuck, that don't make the type 1. And we're sort of deconvoluting more and more. What I want to do is go back and use the ISRAB compound to do a single cell with those to see exactly what's happening. Another question in the live audience. Right. Yeah. Yeah, so in the inflammasome story, we didn't look into the ISR, the ISRIB system. I think partly because some of it, these are shorter time points, but I think it would be interesting to forget about the inflammasome activation, just to with LPS alone stimulation to see what ISRIB does, both in vitro and in vivo models. Also, I think that's something worth thinking about. I have seen some talks where people have tried to link tumor immunity with the ISRIB compound, and I think more to come on that. The one, if that's the last question, I'll just finish with one interesting thing about some of these stress responses. Many of them come during development, right? And it's always just thought that the reason they come up in development is simply to help with the metabolic stress, right? They come on, they come off. But it's conceivable that beyond the metabolic stress, they also come on and off to control cell fate, developmental cues for cell fate and cell function. And so one of the things that I'm really excited about is to do the single cell with the ISRIB. And yes, we'll see lots of metabolic chain genes that were upregulated due to complex 1 loss in the ISR signature, and ISRIB will bring those down. But the real key will be if there are any that are developmental genes, because we are seeing some developmental genes that are being regulated by complex 1 and not by complex 2 as much. There's some differential regulation of developmental genes, but whether they're regulated by the ISR, I don't know. But I think this is something we should think about, that perhaps things like the ISR are regulating developmental programs beyond the metabolic programs to get you through development. Great, we're at 1 p.m. Time for lunch. Thank you.